Hello students, I am Dr. Kishan Kumar Singh and in today's lecture we will discuss about the fat metabolism. Today we will start our first class on catabolic pathways of fat molecules. Food like meats, dairy products, seeds, nuts and oils, they contain dietary fat. Fat in commonly we call lipid also. So in this lesson we will learn about the unique way of lipids broken down, how it will be broken down and how it's absorbed in the digestive tract, and after that how it is metabolized in the cells. So let's start. Fat metabolism is a biological metabolic process. It breaks down the ingested fats into simpler compounds like fatty acids and glycerol, and these simpler compounds undergoes into the cells and produce and process energy for the cells. As you can see in this picture, these are the dietary fats, foods for the dietary fats, and this food uh, undergoes after the digestion it stored in the adipose tissue, and from this adipose tissue this triglyceride is converted with the help of enzyme lipase into three fatty acids and glycerol, and do both get different undergoes in different pathways. We will learn in this class how these things happen in our body. So let's start. The term lipids includes a diverse group of structurally distinct hydrophobic and amphipathic molecules. So the elements of the fatty acids which are quite simple, as you can see in this picture, they have only two part, carboxylic acid and long hydrocarbon chain. It could be 4 to 30 carbon molecules, 12 to 24 is most common. So this kind of chain is could be linear and usually contains even number of carbons. We have seen these things in the previous classes. So the hydrocarbon chain which don't have double bond, we call that saturated, and those contains double bond, we can say that unsaturated. So lipids have four primary functions in the living cells. We have learned these things in previous classes. Like fuel molecules, lipid can be used to store energy, like generally mono, di, and triacylglycerols, and high concentrated energy stores, and signal hormones like steroids hormones. Okay, these are the basically for the steroid hormones. They serve as a signal molecules, and they could become the component of the membrane like phospholipids. Okay, now let's move on to the next slide. So we have learned that carbohydrate is the one of the major fuel for the most all organism, but fatty acids are also a very important energy source. In fasting or hibernating animals or in migrating birds, fat is virtually the sole source of energy. They are the sole source of energy. Um, the body has a limited supply of glucose relative to the energy stored as a fat. So most of the energy in our body is stored as a fat. So we can say that we have limited capacity of storing glucose as glycogen as a source of energy. So about 40% of the body's caloric intake is derived from the lipids, and almost all of these calories come from fats like triacylglyceride. Okay, now let's move on to the next slide. This is fat ingested diet, and it undergoes into the stomach where bile cells act and they emulsify the dietary fats and forming micelles as we have seen in the previous animation. And this intestinal lipase, small intestinal lipase, act as a broken down the lipids into their components, fatty acid and glycerol, and these products of the fatty acid broken down, taken up by the intestinal mucosa and converted into the triacylglycerol. And this triacylglycerol are incorporated with the cholesterol and form apolipoproteins into chylomicrons. Chylomicrons move through the lymphatic system as we have seen in the previous animation and bloodstream to the tissue. This is a chylomicron, and this is the lipoprotein lipase, and they activated the apochylomicron in the capillary and convert triacylglycerol to fatty acids and glycerol. So now let's move on to the this fatty acid enter into the cells, liver cells, and under the liver cells they oxidized as a fuel and re-esterified for storage. Okay. Now let's move on to the next. This is the structure of mitochondria. We have seen previous also. They are double membrane structure, outer membrane, inner membrane, and intermembrane space, where they are filled with the matrix, or where they have contains all of the enzymes for the different fatty acid and amino acid oxidation enzymes. So inside in the mitochondria of the all cells, the enzymes in the outer, uh, that is thiokinase or SL coenzyme synthetase, and inner one is the carnitine, which mediates the movement of free fatty acid derived from the fat molecules into the mitochondrial matrix. In the matrix, each fatty acid molecule exists in the form of active fatty acids or fatty acid coenzyme, and it's broken down into a series of chemical reactions which is called beta-oxidation. It trims two carbons, one acetyl group, and at a time from carboxylic end, beta end, and generating one molecule of acetyl coenzyme in each term of cycle. So we will see this reaction in the later slides. So this animation will show the overall oxidation of fatty acids inside our cell. So as you can see in this picture, this is the mitochondria, and which is the basically site for the catabolism of fatty acids. Brain only able to catabolize short chain of fatty acid, but these organs, liver, heart, adipose tissue, can metabolize long chain fatty acid also. So as you can see in this picture, the only medium and short chain fatty acid can be metabolized in the same. So let's have a quick review. Long chain fatty acids. Long chain fatty acids are the long chain of the fatty acids. They be activated in the outer mitochondrial membrane and transported inside the mitochondrial membrane and oxidized into the matrix. While the medium and short chain fatty acids activated and oxidized in the mitochondrial matrix. Now let's take a quick review on the reactions of beta oxidation of fatty acids. First step is the mobilization of stored fatty acids from the adipose tissue is called as lipolysis. So these are the fatty acid and they lipolysis and they produced triacylglycerols. And these triacylglycerols under converted into the free fatty acids and glycerols, as we have seen in the previous class, with the help of the enzyme lipase. These are the hormone sensitive enzyme. This enzyme is the hormone sensitive, and this glycerol undergoes the phosphorylation and produce glycerol 3 phosphate. And after that, this with the help of enzyme glycerol kinase, and this glycerol 3 phosphate we have seen in the previous classes undergoes the gluconeogenesis. While this free fatty acid move into the bloodstream, they will move into the bloodstream and attach with the same albumin. So this protein will help to go help to go inside the cells. So they undergoes basically in the liver, skeletal muscles, and then not in the brain cells. So this is the fatty acids with the albumin, and they undergoes into the cells as you can see and converted into the fatty acyl coenzyme with the help of fatty acyl coenzyme synthesis. These reaction happen in the cytoplasm, and this is the first step of the uh, fatty acid. They come inside the cell. Now This activated fatty acyl coenzyme A will transported into the mitochondria. Now let's see, it requires 8 1-ATP and acetyl coenzyme A. We know the acetyl coenzyme A. Now this is the outer membrane of the mitochondria and this is the inner membrane of the mitochondria. So this fatty acyl converted to fatty acyl coenzyme A and this is comes the inner membrane. In the inner membrane, you can see the carnitine shuttle. They are inside in the situated at the inner membrane of the mitochondria. So carnitine shuttles transports long chain fatty acids. With the help of this carnitine one transported protein, and they come and attach with the fatty acyl coenzyme A and converted this into the fatty acyl carnitine and release the coenzyme A for further uh, in the cytoplasm for further binding. And this fatty acyl carnitine will undergo into the second of transporter carnitine acyl translocase enzyme, and they undergoes come inside the matrix of the mitochondria. Here we get that this carnitine enzyme 2, carnitine shuttle 2, and they here we release this carnitine free for another reaction, and this fatty acyl coenzyme come into the matrix. So this is the first step basically in which this fatty acid come inside the cell, and this current is and again utilized by the this inner membrane space, as you can see in this picture. Now this fatty acyl coenzyme A undergoes into the beta oxidation. What is beta oxidation? Medium chain fatty acid directly enter to the mitochondria and undergo beta oxidation. But this is for the long chain of the fatty acids. Medium chain could be directly undergo into the beta oxidation. So what is happening in the beta oxidation of fatty acid coenzyme? Now let's see. In the matrix of the mitochondria, there are this is the mitochondrial matrix. As you can see, the oxidation system contains four enzymes that act sequentially to yield fatty acyl coenzyme A, NADH, and FADH2. We will see these reactions step by step. So this is our mitochondrial matrix, and uh, this is step 1 in which acetyl coenzyme A, with the help of acetyl coenzyme A dehydrogenase, convert into the trans-enoyl coenzyme A, and it form one FADH2 molecule. These are the energy carriers which will be utilized by the electron transport chain of the mitochondria. And this trans-enoyl coenzyme A again converted with the help of enoyl coenzyme hydratase enzyme and produce L-beta hydroxy acyl coenzyme. This hydroxy coenzyme again undergoes into the another reaction. Beta produce beta keto acyl coenzyme with the help of this enzyme beta hydroxy acyl coenzyme dehydrogenase, and it to produce a one NADH molecule in this step. One FADH2 in this step 1, and this is the second one, NADH molecule. And this is the third step, fourth step, in which this beta keto acyl coenzyme converted into the acyl coenzyme with the help of enzyme beta keto acyl coenzyme thiolase. Okay, so as you can see, this acetyl coenzyme is produced again. This acetyl coenzyme is free for the binding of another fatty acid molecule. So this is the reaction sequence of the beta oxidation. So we have seen in this in the previous animation that this is the dietary fat of the bird food, and this undergoes into hydrolysis with the help of lipase enzyme in the small intestine, and breaks broke down into the two components, a fatty acid and glycerol. Glycerol undergoes into the phosphorylation and converted into the glycerol 1 phosphate, dihydroxyacetone phosphate, and gluconeogenetic pathway, and finally TCA cycle. While this fatty acid undergoes, it depends on their uh, long chain structure or short chain structure. On the basis of that, it is undergoes into different reaction. The fatty acid, with the help in the mitochondria, this is the go inside the cell with the help of code 36, and this is the cell cytoplasm. Now it's go inside the mitochondria. This is the mitochondrial space and go undergoes in the beta oxidation. We have seen that this reaction in the previous animation also. Okay, now let's move on to the next slide. This slide shows the beta oxidation of fatty acid. It requires different stages. First, the activation of fatty acid in the cytoplasm. Then, this activated fatty acid is transferred from cytoplasm to mitochondria through carnitine shuttle system. And in the carnitine, this mitochondria, inside the mitochondrial matrix, a beta oxidation takes place, which have dehydrogenation, hydration, and second dehydrogenation, then thiolytic cleavage. So this is the first step. You can see that fatty acid is activated with the help of ATP consuming and converted to the acyl coenzyme. This is the first step. This acyl coenzyme A, acyl coenzyme undergoes into the carnitine shuttle system and transported from cytosol to mitochondria. As you can see in this picture, this acyl coenzyme will bind with the carnitine and made acyl carnitine. This acyl carnitine undergoes into this inner mitochondrial membrane and come into the matrix. And this acyl carnitine undergoes again with broken down into two part, acyl coenzyme A and carnitine. This carnitine goes back in the this cytoplasm. Okay. Now this is the beta oxidation of mitochondria. These are the different stages. Dehydrogenation action. This acyl coenzyme A undergoes into dehydrogenation and makes a trans double bond between alpha and beta carbon. As you can see in this picture, here is the energy production step, and they produce energy carrier, different energy carrier. As you can see, first step FAD and FADH2, and then step 3, and then step 4. Fatty acid oxidation could also be divided on the basis of their alpha, beta, and gamma oxy this position into this major and minor pathway. Major pathway is basically for the large molecule of the fatty acid and we known as beta oxidation. However, while the minor pathway is very useful for the partial oxidation of branched chain fatty acid. So these are the different position of the carbon as you can see in this picture. This is the alpha carbon and with the alpha is the that carbon which is attached to the functional group, and this carbon is attached to this carboxylic group, so this is the alpha carbon, and the carbon which attached to the alpha carbon is known as beta carbon, and then so on to omega carbon, last carbon. So this is the fatty acid total different kind of fatty acid pathway, oxidation pathway, major pathway and minor pathway. Now let's move on to the next slide. So if we take an example of palmitoyl coenzyme A, that is 16 carbon oxidation molecules, so how many ATP will be produced? As you can see the number of cycles, and by 2 minus 1 it is around 7 cycles, and so the number of acetyl coenzyme A will be produced n by 2 equal to 8. So 7 NADH as we have seen in the previous. beta oxidation pathway. Each produce 3 ATP. So in the electron transport chain cycle, it will be converted into the 21 ATP. One NADH is equal to 3 ATP, and one FADH is equal to 2 ATP. So 7 FADH is here, so it is equivalent to 14 ATP. In 8 acetyl coenzyme, each provides 12 ATP, so it will be converted into the CO2 and H2A by the TCA cycle, which is around 96 ATP. So total energy yield for the oxidation of palmitoyl coenzyme A, that is 131 ATP. But only two molecules are ATP has been used in the activation of the molecule of the fatty acid. Thereof, there is net yield of 129 molecule of ATP. You can see, so we can see that fatty acid is a huge source of energy. So pathway of the lipid breakdown regulate at three levels. First is the control of lipolysis. We have seen the lipolysis by phosphorylation and dephosphorylation with the help of hormone sensitive lipase. Then it is controlled at the point control of carnitine cycle. Like malonyl coenzyme inhibits carnitine acyltransferase. Thereof, four stop entry of acyl groups into the mitochondria. And finally, it will also be controlled at the beta oxidation point. And like NADH and FADH2 could high production of NADH and FADH2 could also inhibit the beta oxidation. So this is the end of my class. Thank you for the attention. And if you have any doubts, please mail me at my email ID trshell82 at the rate gmail.com. Thank you.